Good morning. This is Ann Brazo with MPN Advocacy and Education International. Twice a year, we try to host a very comprehensive webinar on clinical trial updates. And we reach out to our industry partners so they have an opportunity to share a much longer and much more in-depth presentation on the trials that are happening in their surroundings. And some of the people you will hear from today will not be sharing a lot of that information, but just giving you brief updates on what's happening in their specific organizations. We do this as a way for our patients and caregivers and all the people in the MPN community to really have a firsthand knowledge of what's happening and on behalf of MPN patients. And it's remarkable how many trials are going on. 20 years ago, there were maybe one or two, and now there are just lots and lots of things happening, and the earth is moving under the MPN community. So I'm going to lead off with our first presenter from Insight, Evan Braunstein, who's an MD, PhD, and Senior Medical Director there. Insight has been a huge supporter of ours for many years, and they are the makers of Jakafi. Evan, welcome, and thank you for joining us.
Thank you so much for having me. I will try to share my slides. Is that working?
Yes.
Excellent. So again, thank you so much for having me. It's my pleasure to share some information regarding Insight's clinical trials. Just a brief notice of some information and disclosures. So first, I just wanted to give a little bit of background on Insight for the audience. Insight was founded in 2002, and our headquarters are in Wilmington, Delaware. Over the years, over the past 20 years or so, we've grown from a small company into a bit of a larger company, and now we operate worldwide and have over 2,000 employees. MPNs have always been a focus of Insight, and the first drug approved by Insight was a JAK inhibitor. And because of this, MPNs remain a focus of the company, and we continue to work to improve the treatment of MPNs. These are some of the targets that we are working on in our pipeline, and I'm going to give a brief overview. We are investigating treatment for all MPN subtypes, including ET, PV, and myelofibrosis, and I'm going to briefly review these. Activation of the JAK-STAT pathway is a known driver of MPN disease, and Insight is pursuing treatment of MPNs via JAK inhibitors with both current therapies as well as investigating novel targets. BET proteins are another area of investigation by Insight. BET proteins regulate gene expression in cells, and there's evidence that inhibition of BET proteins can lead to improvement of MPN disease. And so Insight is currently conducting investigational clinical trials in MPN studying this target. The ALK2 pathway is another area of investigation by Insight. Anemia is a common manifestation of disease in myelofibrosis, and inhibition of the ALK pathway can lead to improvement in anemia. And Insight is currently conducting investigational clinical trials in MPN studying this target as well. And finally, Insight is investigating the mutant CALR protein for treatment of MPNs. Approximately 30% of patients with ET and MF have a mutation in the CALR gene that drives their disease. And because this mutant protein is not found in normal cells, targeting mutant CALR may result in suppression of tumor cells while leaving normal blood cells unaffected. And Insight will be conducting investigational clinical trials in MPN studying this target very, very soon. And so that concludes my presentation. I'm sorry that I'm not able to give more detailed information in this forum, but if you would like more information, here are some links and some information to get that information. And again, thanks again for having me.
Thanks, Evan. We appreciate it. And as I mentioned earlier, some of our partners may not be using the full 15 minutes they're allotted because they're able to condense their information and share what they have and what's available. Thanks, Evan. So we are going to move on to Dr. Ray Urbanski and from Pharmacentia. Ray is an MD, PhD, and the Senior Vice President of Clinical Development and Medical Affairs. Are you there? I see your slide, but I don't see you. Ray?
Can you hear me now, Ann?
Yes, I can.
Sorry, fumbling.
No,
around.
that's okay. Thanks for joining us.
Okay. Well, thank you for having me, and I certainly appreciate the opportunity to cover some of the things I'll be covering today. So basically, Pharmacentia has been around for around 20 years, pretty similar to Insight, and they've had a focus on the interferons in general, focusing on not only on the use in hepatitis, which obviously has waned quite a bit with the oral therapies, but also in the myeloproliferative neoplasms. And so that's what I'll focus on today, is really looking at how Pharmacentia actually starting to look at the new possibilities and a new paradigm into how we treat NPNs. So just very quickly, this is the structure of ROPEG and or BESREMI or ROPEG interferon alpha 2b NJFT. And it was purposely designed to basically have a better PK/PD profile than the existing drugs that were used specifically in PV or NPNs in general. And you can see that on the bottom right. When you look at PEG Intron or Pegasus, these drugs can have up to 11 to 14 possible isomers. And why is that important? Well, the PEG moiety that you see on the top right there, that's sort of, I guess, a little greenish, is actually twice the size on a weight-to-weight basis as the actual protein itself. And so one can imagine that because you have this bulky pegylated moiety at the end, that it's going to affect how the drug actually interacts with the receptor, and which leads to its therapeutic effect. So if you have this PEG moiety stuck on 11 to 14 different sites on the protein, you can imagine that you're going to have a very variable and maybe disconnected effect. And that's why the people at Pharmacentia actually developed this site-specific monopegylation. So you get one isomer, the drug does one thing, it interacts with the receptor at the same way. And so at the end of the day, with this site-specific monopegylation, you get a much better safety and tolerability profile, and you get an extended elimination half-life, which for patients means you only have to dose every 2 weeks. And in most of our studies, patients were switched after 12 months of stable counts, actually switched to monthly regimens. So that's the structure of ROPEG. And now let me show you a little bit about, from a mechanism of action standpoint, why we think this is unique as well. So as you can see in a little schematic of a bone marrow, and the pluripotent stem cell is the hematopoietic stem cell. That's the cell that gets mutated and can drive sort of this increased production of blood cells that you see at the bottom there, right? So the erythroblasts, the red blood cells, the platelets, and the white blood cells. JAK inhibitors and other non-selective drugs like hydroxyurea, which is basically a myelosuppressive, will suppress the production of cells below that hematopoietic stem cell. ROPEG acts specifically at that mutated stem cell and therefore reduces the production of the other blood cells by reducing the number of mutated stem cells. And I'll show you some data that will show the dramatic effects of ROPEG or Besremi actually on these mutated stem cells. So it's actually acting at the disease itself and not working further downstream where you're just looking at a sign or a symptom of the disease. So with that, this is ROPEG interferon's clinical trial program. So basically to the left are the studies that have been completed. Most of them have been published or reported out. And these are the studies that did lead to the approval of ROPEG in PV. And I'll discuss those very, very shortly. To the right are the studies that are currently ongoing, the SURPASS-ET, the EXCEED-ET, and the ECLIPSE PV, and I'll show you what they look like and the kinds of the patients that we're looking to enroll and the clinical question that we're looking to address with these particular studies. So we had the PEG and Vera, PROUD PV, and continuation PV, and the continuation PV, Conti as we call it for short, actually was a pre-planned extension of the PROUD PV study. And the thing to bring home here is to notice here is that PEG and Vera, we followed patients for 7.5 years, and for the continuation PV for 6 years. So with a disease where you're going to be treating patients for years, if not decades, having long-term studies and having patients being treated for a long term actually is very beneficial and informative to not only the patient, but the healthcare provider. So with that, let me briefly cover some of the efficacy results that we saw. So this is from the PEG and Vera study. It was a study done in 51 patients, so it was an early phase study. We saw a complete hematologic response rate of 80%. What that means is that your peripheral blood counts actually normalized, and they were below a predetermined threshold. And the time to first hematologic response was about 10 weeks. This varied quite a bit from patient to patient, which is not unexpected with biologics or protein-based therapies. But we did see a rather dramatic effect on the hematologic response. And when we look at this by the various blood counts, so the hematocrit's measuring the red blood cell count, the platelet count, and the leukocyte count, you can see that the platelet count and the white blood cell count goes down quite dramatically. It was basically the effect of ROPEG on the platelet count that led us to really start looking into clinical trials into ET, and I'll mention that in a minute. But you can also see that the hematocrit stays below this sort of this threshold for clinical efficacy of below 45%. So you see good efficacy, but you don't see a lot of thrombocytopenia, or your platelet counts and your white blood cell counts don't go down too low. And your red blood cell count and your hematocrit also does not go down too low. So you don't see a lot of adverse effects when you're looking at things such as cytopenias. And this is a graph that was presented by Dr. Gislinger at the latest EHA meeting. And here you're looking at event-free survival. And event-free survival basically looks at 3 different endpoints, which you can see there: disease progression to either MF or AML, thromboembolic events, and death. And you can see very clearly when it was compared to a hydroxyurea best available therapy arm that there's an early separation, and that separation just continues to grow as you go out in time. So obviously demonstrating the durable effects of Besremi on these 3 composite endpoints. And then more importantly, as I said, the ROPEG acts specifically at the hematopoietic stem cell, the mutated stem cell, and that's usually measured by the allele burden. So the JAK2 V617F, which you see up there, is the most common type of JAK2 mutation. And the allele burden is basically measuring how many cells, how many mutated stem cells you have in your bone marrow over time. And you can see in the green box or the ROPEG arm that the allele burden or the number of mutated stem cells in the bone marrow goes down quite dramatically. When you get to, at baseline, it was around 37% of the patient, 37% of the cells were mutated. When you look after 4 years of therapy, it's now below 10%. And that number keeps on going down as we go out in time. With the hydroxyurea bat arm, you can see that initially the mutated stem cell count goes down, but then it rapidly starts going back up again. And again, Allison Moliterno and others have shown us that hydroxyurea, for example, does cause tumor microenvironment effects in the bone marrow that actually will facilitate or lead to survival advantage of the mutated stem cell, which is why that number goes back up. And certainly you do not want to be on a therapy where your cancer cells start to diminish and then all of a sudden they start to go back up again. So with that, what did all of this data lead to? What it led to the FDA approval in November 2021. You can see some of the things here. I've been in this business a long time. I've never seen the FDA put out a press release like they did, and it's kind of schematically shown there on the right. Once we got approved, you can see a quote from Ann Farrell that's kind of pulled out and bolded from the press release. And basically the key thing here is that ROPEG is indicated for all patients regardless of their previous therapy. So again, another first in my career is that it's not talking about first, second, or third line therapy. The FDA basically approved best remedy across all lines of therapy, i.e. regardless of the patient's previous response or previous therapies. So with that, let me go from the approval now, and I'll move on to the studies that we are currently looking at. So SURPASS-ET is a phase 3. This is an open label, multi-centered study. It's done globally. So we have sites in the US and Canada, as well as in the Asia Pacific region. And it's basically looking at the effects of ROPEG in second line therapy and comparing it to anagrelide. And that is sort of shown here in the study schematic. A couple of things I do want to bring out is if you look at the dosing, which is in the center here, the dosing is different than the dosing that was approved for PV. And this is because we have data to suggest that a more rapid dosing, a higher starting dose and a more rapid titration actually will lead to earlier clinical effects when looking at hematologic response. But we also are hypothesizing that it will also lead to a deeper and more durable response when you're looking at the molecular response or the allele burden. And so this study is currently ongoing. I think we're planning 170 plus patients. We're at 160, I believe, currently. Also note that the dosing is Q2 weeks, but does change to Q4 weeks once the patient has been stable for at least 12 months. These are the IE criteria. I won't go into this in any great detail, but it's any subject who was diagnosed with high-risk ET, and they have a JAK2 V16F positive mutated stem cell, as I mentioned previously, and they have to have documented resistance and tolerance to hydroxyurea. So as I mentioned, this is a second-line study. Here is the information. I'm sure Ann will have these on her site, so you can certainly pull them up if you're interested in joining this study. So that's the SURPASS-ET. Now this study, the EXCEED-ET, is a single-arm study, and this study is only being conducted in the US and Canada. And this is to ensure that we demonstrate drug activity with a population that's representative of those in North America, since the majority of patients in the SURPASS study are being recruited outside of the US. So it's a single-arm, multi-centered study, as I mentioned, and it's looking at patients with ET. This is the study diagram. So again, diagnosis based on the 2016 WHI criteria. And basically, we're including anybody that requires cytoreductive therapy. You could be cytoreductive naive, and your physician seems to think you need cytoreductive therapy or disease-modifying therapy, I should say. Or you could have been on hydroxyurea or anagrelide in the past. And so here's the schema. And again, this study, like SURPASS, is utilizing a different dosing than what's in our label of a 250 starting dose and migrating or titrating, excuse me, to a targeted dose of 500. The primary analysis is going to be at week 56, which you see here. And then we're going to follow patients outward because the FDA, as well as us, are very interested in the durability of response with ROPEG with these ET patients. So the population is quite different than the population we're studying with SURPASS. So we will have, very much like we did with PV, have encompassed or incorporated basically every patient and patient phenotype that you would see with PV or ET. These are the major inclusion and exclusion criteria. I just want to make a point that we are currently putting in an amendment that you do not have to be interferon treatment naive. You could have been on previous interferon treatment, but you just could not have had an SAE, a serious adverse event, or basically been resistant to therapy. So if you have been treated with a prior interferon, you could still get into the study. And you can see the major exclusion criteria here as well. Primary and secondary endpoints. Again, the primary endpoint is a durable response rate. Looking at the hematologic responses, very much like I showed you previously. Looking at the platelet count and the white blood cell count. We are also going to be looking at the clinical response by ELN criteria. And as I mentioned, durability of response is incredibly important. We are looking extensively at the genomics, looking at the changes in CALR, MIPL, or JAK2 allele burden over time. This is incredibly important because, as I said, based on the mechanism of action of ROPEG, we believe that it actually treats the disease at the source of the disease, meaning that mutated stem cell. And so we fully anticipate and expect to see these mutated stem cells, regardless of what is driving them, actually diminish over time with ROPEG treatment. So it's approximately 40 centers across the US and Canada. As I mentioned, 64 patients will be enrolled, and hopefully we'll get 56 to complete. You can see very briefly here a 28-day screening, titration, and then a 1-year follow. And we will be following up these patients over time. Here is all the contact information should you need it. And as I said, these slides most likely will be posted. And last but not least is the Eclipse PV. This is a study. It's a phase 3B study. It's a randomized, it's a parallel group study. And it's going to be addressing a very important question. I did mention to you previously that the dosing that we're using in ET is different than the dosing that's in our current label. So this study is actually going to address, is there a difference between the time to reach primary endpoint, the depth of the endpoint response, as well as its durability. And this is both for CHR as well as the molecular response. So this is a 2-arm study comparing the labeled dosing, as you can see on the top here, and we're comparing it to the more rapid dosing that I've been showing you with the ET trials. And so this is going to answer a very important clinical question based on what happens to your hematologic rate or blood counts in the periphery, and then what's going on in the bone marrow, i.e., are we really driving down that allele burden or really decreasing the number of mutated stem cells in your bone marrow by utilizing this more rapid and higher dosing titration. These are just some of the inclusion exclusion criteria. Again, we tried to emulate this quite closely with what we did in our pivotal studies for the PV indication. So you can see the criteria here. It's really not that restrictive at all, and you can see the main exclusion criteria. Again, it's very typical and not exclusionary. These are the primary endpoints. The CHR at week 24. So we want to see by month 6 how rapidly the different dosing paradigms actually led to a hematologic response. You can see the other secondary endpoints that I have listed here. Again, we are really interested in looking at that JAK2 allele burden over time and any change in any other non-driver mutations. So what are we doing with these different dosing paradigms to these mutated stem cells that are really driving the disease phenotype? And my last slide here is just basically to show you that we're doing approximately 30 centers across the US and Canada. We're looking at about 100 patients. You can see the schema in bulleted form up on the top right there with screening periods, titration, and then the core treatment period and the extension. And any additional information will be found on the slide here. So, Anne, that's all I had. So thank you very much.
That was really, really interesting. Thank you. And I'm assuming in those links that a patient or a physician would be able to find out where these locations are across the US and Canada. I mean, where you want to ramp up and what areas.
That's correct.
Okay, all right. Thank you very much, Ray. I really appreciate it. That was fascinating. I'm so glad you're looking at ET now because we have so many ET patients that need some help. We will move on to Imago Biosciences, which is now a subsidiary of Merck and Company Incorporated, and Alicia Shilton Lloyd, who is a PhD and the Vice President of Medical Affairs. And then she will introduce her colleague, who is the clinical director. Alicia?
Hi, can you hear me okay?
Yes, I can.
Perfect. Hi, thanks so much, Anne. As Anne said, I'm Alicia, and I head up medical affairs and the inpatient engagement team at Imago Biosciences. Imago is a subsidiary of Merck & Co. Incorporated, and it is a clinical stage biopharmaceutical company developing new medicines for the treatment of myeloproliferative neoplasms, MPNs, and other bone marrow diseases. Our lead candidate is bomedemstat. Bomedemstat is an investigational once-a-day oral lysine-specific demethylase 1, or LSD1, inhibitor. LSD1 is believed to play an important role in the maturation of blood cells in the bone marrow. Bomedemstat is an investigational agent, which means it's not yet approved by the FDA for use outside of clinical trials. Over 200 patients have been treated with bomedemstat in multiple phase 2 clinical trials for the treatment of essential thrombocythemia, myelofibrosis, and polycythemia vera, in addition to other indications. We presented our latest data on these trials in December at ASH, which is the large annual conference here in the US, where the latest data on blood cancers and other blood disorders is often shared. I'm now going to turn you over to Dr. Uzar Obu, MD, PhD, Clinical Director of Oncology for Merck Research Laboratories. Dr. Obu is the clinical director responsible for our currently enrolling PV and upcoming ET trials, and will share with you the data we presented at ASH, as well as provide an update into our clinical program overall. Over to you, Uzar.
Thank you, Alicia, and I'm happy to be here today to share our initial results, or some of the results we shared at ASH with everyone. Let me get my screen sharing up. There we go. So again, I'm happy to be here to speak to you today and share some of the results that our Imago colleagues have produced using bomedemstat, our LSD1 inhibitor. We're very excited that Imago is joining the Merck family, and I think we're even more excited about where bomedemstat can go. And on top of that, we're excited to continue the collaboration Imago has had with you in directing what's important to our MPN patients and what you think we should be doing with this agent. This one is, as Alicia pointed out, is an LSD inhibitor. The mechanism of action is a little different from what you've seen before. I will start here by talking, so from the patient perspective, what do we think this can do from a mechanistic rationale? And if you look at the far right-hand side there, you see that by inhibiting LSD1, we think we can reduce constitutional symptoms and bone marrow fibrosis, which would translate to reducing fatigue, anemia, pain, itching, night sweats, and also reducing the need for a reduction in splenic volume among patients who have an increase in spleen due to one of the myeloproliferative neoplasms. We also, as some of our previous presenters have mentioned, have seen that it does reduce the mutant hematopoietic stem cell, which would reduce the allele burden, which would then be a disease-modifying effect. And I'll talk about some of the results we've seen initially so far. For the more clinicians involved, or those that are more into the details, you can look there from going from left to right. What is LSD1 inhibition doing? We're reducing the inflammatory cytokines. We're reducing the number of platelets. In this case, we're looking at ET patients. And also by reducing the number of growth factors, we reduce the reticulin and collagen deposition, which is what is leading to bone marrow fibrosis. And obviously with reducing platelets, we're also reducing thrombosis and bleeding. I will talk a little bit about one of our ET trials. So this was a phase 2 ET trial in patients that have received prior therapy. This trial is no longer enrolling patients, and it was presented at ASH, as previously pointed out. This trial enrolled 73 patients. The patients were between the ages of 42 and 92, was a mix of males and females, about 42% male. 33% of those patients had a history of bleeding, and 18% had a history of thrombotic events. As you would expect, most of them had a JAK2 mutation, 49%, but CALR and MPL mutations were also represented here, the three main driver mutations. We also had patients that were triple negative, so they didn't present with any of these three mutations. And we had patients that had other mutations, in potentially on their own or in addition to the three main ones. The total symptom score for myelofibrosis, which some of you may be familiar with, is there represented. It was a range from 0 to 74 for patients enrolled on this trial. 84% of them had been treated previously with hydroxyurea. 63% had received one or more prior cytoreductive therapy, most likely hydroxyurea, and 37% had received at least two prior cytoreductive therapies, and there was a maximum of 4. In that lower box there, you see the total range of therapies that patients had received. So hydroxyurea in 70% of them, anagrelide, interferon, busulfan. So they had all received all of these patients before, all of these therapies, or one of these therapies before coming onto this trial. If we look at this from a safety perspective, we can conclude bomedemstat, based on the trial, what we've seen in results so far, that it's generally safe for patients with ET. Most of the AEs or medical issues that they experienced were mild, with the most common one being dysgeusia or a change in taste. And this change in taste is really a case of where sweet things tend to taste sweeter. It's time limited, and we're able to manage it with sort of timing of dosing, and it goes away after a while. The table on the right is a little busier, but it shows you the total range of adverse events that occurred at any point for patients that were on the trial, and supports our conclusion that most of these events were mild, which is grade 1 or grade 2. You see the grades for 3 or 4 are pretty low percentages all around. Dysgeusia, as you can see, is the most common one, but there was constipation and thrombocytopenia. Some of this thrombocytopenia this is a phase 2 where we were, figuring out how to dose, how to escalate. Sometimes it went up a little bit, and we have a better sense of what to give, how far to escalate. So we don't expect to see thrombocytopenia going forward as one of the more common AEs. Moving on to what we saw from a results perspective. Sorry. So just looking at it here, at 24 weeks, 95% of patients had received a hematologic response. At the time of analysis, 28 of those, of the 28 patients who reached the 48-week duration, 89% of patients had a durable response. So this is having a reduction of your platelets into the target range and having no incidence of a major bleed or thrombosis for a duration of at least 12 weeks. If you look on the right there, you see the average platelet count for patients that were enrolled on the trial, for 73 of them. At screening, which is the very first tick you see there, most of them are sort of between that, about 700,000. And you see that reduction going down over time on treatment, at about 12 weeks, almost all patients had gone into the target platelet range, which is less than 400. You also see in the lower two graphs I have there on the right that there is very limited effect on your white blood cell count and limited effect on hemoglobin. So we're not going to see the sort of cytopenias that have been seen in some of the other treatments available to date. I did mention about the allele burden, reducing the allele burden, and you see that there on the figure on the left. There's a 67% reduction in the allele burden. I had pointed out the types of mutations that were there, and all patients across the range of mutations, the JAK2, the MPL, the CALR, the patients responded regardless of the type of mutation that they had. Going on to looking at what does it mean in terms of patient symptoms. There is 75% of patients had a reduction in their worst symptoms over time while taking the drug. You see there, based on the TSS score, bone pain, there was a reduction, average improvement in bone pain, and an improvement in fatigue. And not listed here, but there's also improvement in itching, which were some of the ones just to highlight. So patients overall had an improvement in their symptoms while taking this drug, beyond sort of the biochemical changes of getting the platelets down into the right range and not experiencing the cytopenias, as I mentioned. Shifting gears slightly here, we want to talk about the initial trial in myelofibrosis, which is the MK3453-002 trial, which is also currently close to enrollment. But 89 patients were treated on this trial. Their ages range from 35 to 88. They were pretty much split between male and female. Here we had a mix of patients who, and sorry, there's an error here in the disease subsite. We had a mix of patients who had primary myelofibrosis, post-ET myelofibrosis, and post-PV myelofibrosis. 53% of those patients were high risk. Here we have the spleen volumes there. I'll focus a little more on the mutations. So JAK2, 64%, CALR, 23%, 7% had MIPL, and again, about 6% were triple negative, and then there was a mix of other high-risk mutations listed there. In terms of prior therapies, there was a median number of 2 prior therapies, but we had a range of patients who were treatment naive to those that had received at least 6 prior therapies. And you see there a list of 73% of the 73 of the patients of the 89 received ruxolitinib, 12 received anagrelide, 3 received fedatinib, and 42 had received hydroxyurea previously. Again, shifting to an initial look at safety with the same conclusion that it's generally safe and tolerable. The most common AEs or adverse events or medical condition experienced for patients on the trial were thrombocytopenia and a change in taste. Again, the change in taste seems it's a common theme here. It is, again, making things sweeter. It's time limited and manageable. The thrombocytopenia, again, is in a setting of where we were trying to figure out what is the right dose, how are we escalating, and what's going on here. So we figure that out, and we don't expect thrombocytopenia to be a significant issue going forward. To talk a little bit about what have we seen coming out of this. Of the patients that were on the trial, 59 of them had had their bone marrow evaluated, and 85% of those patients had shown either an improvement or stabilization in their bone scarring. 66% of patients had a reduction in their spleen volume, and of those, overall 28% had a reduction in spleen volume of 20% or more. And you see this represented in the, these two features represented in the figures on the far right there, with the green column showing a change in the myelofibrosis, this grade for scarring, the green one showing a reduction, the blue showing stabilization, and some patients unfortunately did have an increase. In the lower chart there, you see a change in splenic volume. It's a little small to see, but overall there's a vast reduction in splenic volume for patients while they were on this. Importantly, we did see a decrease in the allele frequency, and we had no patients progressing to AML while on bomedemstat. Thinking about what does this mean for patients? If we talk about blood transfusions, 52% of patients had a reduced need for transfusions or a stabilization in their need for transfusions. And of those, 3 patients had no need for transfusions any longer. If we look at symptoms, 65% of the patients had a reduction in their worst symptoms or had improvement in their worst symptoms over the course of treatment. And of those, 19% of patients who had significant symptoms had an improvement of 50% or more. In a nutshell, that is what we have seen so far with our initial phase 2 bomedemstat trials in myelofibrosis. We do have a trial open now exploring bomedemstat in polycythemia vera. We are planning to do a follow-on study for myelofibrosis and ET, as those were phase 2 studies. We're planning on treating more patients and hoping to replicate those results and have that available. Again, Merck is happy to welcome the Imago family and the MPN community, and we'd love to hear from you and get your feedback on what you think we should be focusing on.
Thank you so very much. That was excellent. And again, I just can't help reiterating that our patient community is extremely happy to know that these drugs are coming down the pipeline, and that the trials for ET as well are a long time coming. So this is really great news. Thank you very much.
Thank you.
So we'll move on to Jessica Forrest and GSK from GSK, and she is the patient engagement liaison. Hello, Jessica.
Hi, Anne. Hi, everyone. Thank you so much for having me today.
Yeah.
I have a very brief overview, a brief update here. As the patient engagement liaison, my role is a lot less clinical. It's much more dealing with education with patients. So just a very brief update here, but I'm really appreciative for the opportunity. So thank you so much. So a little bit about GSK. GSK is a global biopharma company with a purpose to unite science, technology, and talent to get ahead of disease together. GSK is focused on maximizing patient survival through transformational medicines. Our focus includes immuno-oncology, cell therapy, tumor cell targeting therapies, and synthetic lethality. Our goal is to achieve a sustainable flow of new treatments based on a diversified portfolio of investigational medicines, utilizing modalities such as small molecules, antibodies, antibody drug conjugates, and cell therapy, either alone or in combination. In July of 2022, GSK completed the acquisition of Sierra Oncology, a California-based biopharmaceutical company focused on targeted therapies for the treatment of rare forms of cancer. The acquisition of Sierra Oncology includes the molecule momelotinib, a late-stage potential new medicine with a unique dual mechanism of action that may address the critical unmet medical needs of myelofibrosis patients with anemia. In August of 2022, GSK announced that the FDA accepted the new drug application, or NDA, for momelotinib. If approved, momelotinib will contribute to GSK's growing specialty medicines business with a US launch anticipated in June of 2023. The NDA is based on the results from key phase 3 trials, including the pivotal MOMENTUM trial. The primary analysis data from the MOMENTUM trial was recently presented at the 2022 American Society of Clinical Oncology annual meeting and the European Hematology Association 2022 Hybrid Congress. To close, I'd just like to add that we are so grateful to Anne and the entire myelofibrosis advocacy community for all the work you're doing to raise awareness and educate patients and their care partners about this disease. We appreciate the partnership that we have all have with your MPN Advocacy and Education International, and I thank you for this opportunity to tell you and your listeners about what we are doing at GSK Oncology. Thank you all so much.
Thank you very much, Jessica. We really appreciate it. And we know some companies, are not at the place like others
Sure.
to share extensive clinical trial information, but we appreciate it. And I'm glad you were here today.
Thank you. Thank you for the opportunity. Have a good day, everyone.
Yes, you too. And so we'll move on to CTI Biopharma and Sharon Martin, who's the Senior Medical Science Liaison. Sharon?
Good morning. Let me just share my screen here. And are you seeing just the slides or the notes?
I'm seeing the slides and the side of the other slides.
Okay. Well, it's not giving me the option. Let's see. Usually there's a drop down. I am so sorry.
It's all right. Would you like to figure that out and we'll go on to
Yeah, that'd be great. Thank you.
Okay, we'll move on to let's see, morphosis, and Stephanie Goldbaum, who's an MD, MA, Director of Medical Affairs. Are you with us, Stephanie? Who do I have with me? This does happen. Technology isn't always so great. Oh, there you are, Stephanie.
Yes, I'm here. Thank you.
Yes, I will hand this over to you.
Sorry, I think we are actually also having the same problem with the slides.
So you're just sharing your screen. So you open your slides and then just share your screen, and it should not show all of this stuff. There you go. You've got it. Okay. Thank you, Stephanie.
Well, thank you so much, Anne. Really appreciate it. As I think you mentioned, I'm Stephanie Goldbaum, Medical Director of Collaborative at Morphosis. It's a real pleasure to be here today. And thank you to the NPN Advocacy Education International for their partnership and this opportunity to share a little bit about MorphoSys. So for those of you who aren't familiar with MorphoSys, I'll start by giving a brief overview of the company. So MorphoSys began as a research and development technology provider over 30 years ago in Germany. Now today, we've evolved into a fully integrated commercial stage biopharmaceutical company based both in Munich, Germany, as well as in Boston, Massachusetts, and we're focused on the development and delivery of innovative cancer medicines. Our work doesn't stop when the research is done or the discovery made. Now, the burden of cancer is vast on patients, on their families, and on healthcare providers, but so are our aspirations to redefine how the disease is treated. Now, MorphoSys' pipeline consists of investigational cancer medicines in mid and late stage development. Now, this includes pelabresib, our investigational BET inhibitor. It's currently being explored as first-line treatment. It's a first-line treatment option for patients living with myelofibrosis. We are studying pelabresib in combination with a JAK inhibitor called ruxolitinib in both phase 2 and phase 3 clinical trials. Now, those clinical trials are called the MANIFEST and the MANIFEST-2. Pelabresib is an orally administered small molecule. So in other words, it's a pill designed to inhibit the activity of a family of proteins called bromodomain and extraterminal domain proteins, otherwise known as BET. These are called BET proteins. So BET proteins can contribute to abnormal gene expression in myelofibrosis. And inhibiting these proteins does really represent a potential therapeutic approach to the treatment of myelofibrosis. Now, as a reminder, pelabresib is an investigational new drug not approved for by any regulatory authority. There is no guarantee that it will be approved. So a little bit about our MANIFEST-2 trial. So MANIFEST-2, it's an ongoing phase 3 trial, and it's studying the efficacy and safety of pelabresib in combination with ruxolitinib, a JAK inhibitor, versus ruxolitinib alone in patients with myelofibrosis that have never been treated with a JAK inhibitor before. So these patients are JAK naive patients. MANIFEST-2 is a global, multi-center, double-blind study of more than 400 patients, and they are all JAK inhibitor naive patients. And just to let you know, in case you don't know that you've never heard double-blind study before, a double-blind study is when neither the patients nor the researcher knows the treatment that the patient is receiving until after the study is completed. There are over 400 patients in this trial with myelofibrosis. Patients in this study are randomly assigned to one of two groups. So they either receive pelabresib and ruxolitinib or ruxolitinib plus placebo. MANIFEST-2 has recently reached full enrollment and is no longer recruiting patients. So now for the primary endpoint, I'm just switching. Okay, so here we can see the primary endpoint. The primary endpoint of a clinical trial is the main result that is measured at the end of the study to see if a given treatment has succeeded. So the primary endpoint in the MANIFEST-2 trial is the proportion of patients who receive a 35% or greater reduction in spleen volume at 24 weeks of treatment in the clinical trial. And this you may have heard of SVR35. It's referring to the same thing. So reduction in spleen size is an important clinical endpoint in myelofibrosis because spleen enlargement may correlate with disease activity as well as it may cause significant pain and discomfort. Now, the secondary endpoint in this trial, or secondary endpoints in general, actually may provide supportive information about the therapy's effect on the primary endpoint, or it may demonstrate additional effects on the disease. So in the MANIFEST-2 trial, the key secondary endpoint is a proportion of patients that achieve a 50% or greater improvement in the total symptom score. as measured by the myelofibrosis symptom assessment form version 4, and that's 24 weeks after starting treatment in the clinical trial. As you may know too well, people living with myelofibrosis may experience a severely diminished quality of life due to symptoms such as fatigue, fever, weight loss. The myelofibrosis system assessment form is a validated self-assessment tool designed specifically for myelofibrosis patients that can track changes in these symptoms. And as you can also see on the slide, we are looking at the bone marrow fibrosis as well, and we're determining how much improvement in bone marrow fibrosis is seen at week 24 compared to baseline. So just to summarize, pelabresib is an investigational oral small molecule drug designed to inhibit the BET proteins. Now we know BET proteins regulate key pathways involved in myelofibrosis. So inhibiting the activities of these proteins represents a potential therapeutic approach for myelofibrosis. We are studying pelabresib in combination with a JAK inhibitor, ruxolitinib, in both a phase 2 and a phase 3 trial named MANIFEST and MANIFEST-2. And a phase 3 MANIFEST-2 trial aims to evaluate the efficacy of pelabresib and ruxolitinib versus ruxolitinib monotherapy in JAK inhibited naive patients with myelofibrosis. Now, as I mentioned, enrollment is complete as of April 2023, and the data is top line data is expected at the end of 2023. So we are committed to people living with myelofibrosis who need options for first-line treatment and beyond. At MorphoSys, we are honored to work closely with patient advocacy organizations serving the blood cancer community, including NPN Advocacy and Education International. We engage with the community through forums such as this one today to understand the patient perspective and journey so that we can incorporate those critical insights to our research and to our development efforts. Thank you again for your partnership.
Thank you for your partnership, and thank you. That was really interesting. I appreciate it. And I'm sure our viewers appreciated that. Thanks very much. Let's see if Sharon Martin is available. Should we try it again, Sharon, with CTI?
I think we're good.
Okay.
Let's go for it here.
I'm going to go away and I'll make sure I let you know if it's not showing up.
All right. Are you just seeing?
Perfect.
Slide. Okay. Second time's the charm. I appreciate your patience. And I want to thank you, Anne, and your team for hosting this incredible educational event and the opportunity to provide a quick overview of CTI Biopharma and the update and an update on the work that we do. So CTI is based out of Seattle, Washington, and we focus on developing novel therapies for patients with rare blood-related cancers through a better understanding of the disease drivers so that our therapies can hopefully better meet the evolving needs of those who are actually living with the disease. As many of you know, patients with myelofibrosis specifically, who also have cytopenias or low blood counts, have historically had very few therapy options. We also know that low platelets, low hemoglobin, and a big spleen are all independent prognostic risk factors to consider when treating patients with myelofibrosis. The development of the first JAK1, JAK2 inhibitor was a breakthrough for the community, and especially for those whose lives were impacted by the symptom burden of the disease. And over the last 10 years, we have learned so much more. There's now an even deeper understanding of the biology that drives myelofibrosis. We now have a better understanding of the inflammatory pathways that cause the spleen to enlarge, that then cause the life-altering symptoms like fatigue, bone pain, and itching, as well as low platelets and anemia. Driven by our mission, CTI is making a meaningful impact on the lives of patients with MF and cytopenias. Just last year, the FDA granted accelerated approval for pacritinib, a JAK1 sparing JAK2 IRAK1 inhibitor for patients with myelofibrosis who have very low platelet counts, meaning less than 50,000, because these patients often have more advanced disease, and many can only tolerate a fraction of a dose of currently available treatments. Pacritinib, which is marketed as Vonjo, is an oral drug taken twice daily, and it's indicated not only in the frontline setting for use in patients with platelets less than 50,000, but the National Comprehensive Cancer Network, or NCCN guidelines, also include pacritinib as second-line therapy with no upper platelet limit after another JAK2 inhibitor is used. Pacritinib is also recommended for lower risk MF patients who have lost or not achieved symptom burden response. As I mentioned, over the past 10 years, much more has been learned about myelofibrosis and the different pathways that drive the disease. And so, pacritinib has a slightly different mechanism of action versus the early JAK inhibitors. While sparing of JAK1 or not hitting JAK1 may explain the lower rates of infection and hematologic stability we see with pacritinib, IRAK1 is also an important target in myelofibrosis because when you inhibit IRAK, you also inhibit the NF-kappa B pathway, which is another driver of myelofibrosis, inflammation, and disease progression. You can also see in the gray box on this slide, and this is really important, that the half-life of pacritinib is quite a bit longer than the other JAK inhibitors, which means it may take a little longer to reach full efficacy. Continued interest in the unique mechanism of action of pacritinib recently led to a retrospective review of data from the PERSIST-2 study, which was the pivotal phase 3 trial that led to FDA approval. In December of 2022, Dr. Stephen O presented that data at the American Society of Hematology meeting, or ASH. This data assessed pacritinib's impact on another important pathway in myelofibrosis called ACVR1, which is a receptor kinase that plays a role in anemia. Data showed that pacritinib is a very potent inhibitor of ACVR1, in addition to having JAK2 and IRAK1 inhibition. This unique kinase inhibition profile for pacritinib potentially offers a unique mechanism for the improvement of anemia. The data also showed that a significantly greater proportion of patients who received pacritinib compared to best available therapy achieved red blood cell transfusion independence over any 12-week period through week 24. So turning to our ongoing clinical trials, the Pacifica trial is our phase 3 confirmatory trial currently enrolling outside the United States. The Pacifica trial inclusion criteria includes patients with primary or secondary myelofibrosis who have a platelet count of less than 50,000 and have had limited prior exposure to another JAK inhibitor. It's looking at the efficacy of pacritinib 200 milligrams BID versus physician's choice, which does include low-dose ruxolitinib, and has co-primary endpoints of spleen volume reduction and total symptom score reduction. Again, it's actively enrolling just outside the United States. Finally, I'd like to share with you some very high-level information about our investigator-sponsored trial program. As you can see on this slide, we currently have 3 hematology-focused trials, 2 phase 1/2 trials looking at pacritinib's utility in graft versus host disease, which is a complication that can occur after stem cell transplant, 1 being led by Moffitt Cancer Center and 1 being conducted at the NIH. We also have a phase 2 investigator-led multi-center study from the University of Michigan looking at pacritinib in relapsed refractory T-cell lymphomas. And if we switch gears to solid tumor trials, we do have 1 ex-US investigator-led study looking at pacritinib in patients with 1q21 amplified solid tumors, and 1 study being conducted at the Medical College of Wisconsin looking at pacritinib in advanced prostate cancer. In conclusion, CTI Biopharma is committed to ongoing support of rigorous science and data so we can bring much needed treatment options to the patients living with MPNs and other cancers, with the goal in myelofibrosis of improving symptoms, spleen response, and providing hematologic improvement or stability. We've included a couple of links here where you can find more information on our active clinical trials, and thank you again for the opportunity to be here with you today.
Thank you, and that was very well done, Sharon. Thank you. We appreciated that. We are moving on. We're actually moving on pretty quickly here. So I will check to see if Protagonist is with us. They're not here yet. Steve Romano from Silence. I'm pretty sure I just saw you. There you
Yeah,
are.
yeah.
Would you mind going next? And Steve is an MD and the incoming head of R&D and Chief Medical Officer at Silence Therapeutics. They are new to us in the last year, but very, very agile, and we're really looking forward to hearing what Steve has to say. Thank you.
Yes. So
Here, I'll keep my microphone on. I don't know what happened here.
okay, here we go.
There
Can
you go. Share your screen and perfect.
Beautiful.
Thanks,
Okay.
Steve.
Thank you so much. And thanks for your patience in getting this set up. So yeah, I'm Steve Romano. I'm the Chief Medical Officer here at Silence Therapeutics, and I'm delighted to talk with you guys today and tell you a little bit about our company, our technology, and also a couple of the programs that we're running, one in particular of interest to this group. So we're a global biotech company. We've got our company headquarters are located in London. We have our research and development operations in Berlin, and we have offices in the US in New Jersey, and that's where I'm sitting today. And we've got about 125 employees worldwide. So our technology is a proprietary small interfering RNA platform. We refer to it as the GOLD platform, and it's associated with post-translational gene silencing. Now, as a quick reminder, and I know this group knows this, but the process by which DNA is copied to RNA is, of course, transcription. The process by which RNA is used to produce proteins is called translation, and our siRNAs interfere with the latter. So our platform allows us to discover and develop precision engineered medicines that target specific conditions by silencing disease-associated genes. Now, each siRNA is uniquely designed and can only bind to a target messenger RNA, which can reduce the potential for side effects. They have broad utility. We're targeting both rare and common genetic diseases, and I'll talk about both in a moment. It's a durable effect, but it's reversible. So siRNA therapies can achieve a long-lasting effect after even a single dose and without permanently altering the gene. And we're advancing a pipeline of wholly owned clinical programs in cardiovascular as well as rare diseases. And we have 14 or so partnered programs as well. So these are programs that we're working with other companies on, and they're taking advantage of our technology. So to our clinical programs, we've got two programs right now that wholly owned that have reached the clinic. The first is zerlasiran, and I'll only mention this briefly. It's targeting LPA. Now, LPA is a type of LDL, and it's genetically determined and associated with an elevated risk of cardiovascular disease. Unlike LDL, though, it's not amenable to, for instance, diet and exercise. So it really requires new medicines to target this kind of residual risk associated with an LPA elevation. And this is just a testament to our technology. We did a phase 1 study in 32 healthy volunteers. We saw a really robust reduction in Lp(a) down to about 98%, which persisted even after 5 months of the observation period, and it was very well tolerated. So we're very excited about that. That's moved into phase 2 and looking forward to providing more data sometime next year. Now, the one I really want to talk to you about is SLN124, and this targets TMPRSS6. Now, TMPRSS6 is a gene that provides instructions for making a protein, matriptase 2. Matriptase 2 itself is part of a signaling pathway that controls hepcidin. And, we want to remove the inhibition that TMPRSS6 plays on that pathway so that you can, of course, have an increase in hepcidin and potentially modulate some of the blood conditions that we're interested in evaluating. So let me tell you a little bit more about that. So SLN124 is our compound targeting hematologic disorders. It targets TMPRSS6, as I mentioned, and modulates hepcidin. Hepcidin, as you probably are familiar with, is the body's master iron regulator. We have very nice positive preclinical data that showed therapeutic potential in several different blood disorders. And we've established proof of mechanism demonstrated in healthy volunteer study. Now, we've also been able to obtain a number of regulatory designations, which we hope will allow us to sort of facilitate or speed the development of this product for many of the conditions we're interested in, if in fact the data hold up. We have fast track designation for polycythemia vera, PV. We've got orphan drug designation for PV, beta thalassemia, and myelodysplastic syndrome. And we have a rare pediatric disease designation for beta thalassemia. Now, as I mentioned, we established proof of mechanism in healthy volunteer study. That study was referred to as Gemini. That showed efficacy, durability, and safety. That data was presented at ASH back in December 2021. So that was about a year and a half ago, and that's accessible. So let me just show you a couple data slides from that particular presentation. So here we see that SLN124 increased the average hepcidin up to 4-fold after a single dose with activity sustained for about 2 months. And as you see here across the bottom, you have a group of bars for placebo for SLN124, 1 mg per kg per body weight, 3.0 mg per kg, and 4.5 mg per kg. Obviously, placebo had no relevant effect on hepcidin levels, but you can see a really nice dose effect as you go from 1.3 to 4.5 on the other graphs. Each of those points on the graph is a different timeline. So baseline, approximately 1 month and approximately 2 months. So we've got a nice increase that is dose related, and you see a persistence of that effect over the course of the observation period. Now, of course, we're also interested in the downstream effects of that. So is the rise in hepcidin having an impact on other important parameters for iron metabolism? And yes, we're seeing that that's the case. So we've seen a reduction in serum iron by about 50% in this study after a single dose, and again, with activity sustained for over 2 months. And again, the same set of slides, the same set of data. To the far left is placebo, and you see no relevant change in iron levels. But as you progress to the drug doses of 1, 3, and 4.5, you're seeing a nice reduction across each of these. Now, remember, these are in healthy volunteers. So we have a good clinical rationale for applying this to polycythemia vera, which is a very interesting area. As you all know, polycythemia vera, or PV, is associated with genetic mutations, which cause overproduction of cell lines, in particular red blood cells. That high hematocrit is increasing blood viscosity or thickness and contributes to elevated thrombotic risk or clots. Patients with hematocrit above 45 to 50% are about 4 times more likely to die from cardiovascular causes or major thrombotic events than those who maintain their hematocrit below 45%. So most PV patients are also iron deficient at diagnosis, and repeated phlebotomy exacerbates this. Of course, that's a standard of care for many of these patients. Preclinical data that demonstrated the hepcidin modulation of PV is now published in Blood, and I'll leave the journal reference here for future use. And SLN124 elevates hepcidin, leading to the blockade of iron release to the circulation, restriction of supply to the bone marrow. So iron in the bone marrow, of course, is essential for red blood cell production. Restriction of this cofactor reduces RBC generation and reduces hematocrit, and our hope is to demonstrate that that has a clinical impact on the important outcomes in patients with PV. Because of that, we're moving forward, and we've just initiated a study, a phase 1 and 2 study in combination in polycythemia vera. The name of that study is SANRECO. That's a funny name. San derived from the Latin for blood and RECO for recovery. We're including adult patients with PV, and we're assessing safety, tolerability, efficacy, and of course, pharmacokinetics and pharmacodynamics. And it's administered by subcutaneous injection. So the phase 1 is an open label portion, and that's really a dose finding phase. And we hope to quickly identify an optimal dose to bring forward in a seamless way into the phase 2 portion of the study, which is a randomized placebo-controlled double-blind trial. The primary outcome measures are, of course, evaluating the safety and the incidence of treatment adverse events, but I think more importantly, the assessment of the number of phlebotomies at the various intervals that we're observing these patients for. So we would hope that the drug will be effective at reducing the requirements for phlebotomy. And as I mentioned, we'll also evaluate PK and PD parameters. Now, in conclusion, SLN124 is safe, has a long duration of action. Long duration of action from a single dose predicts a long interval between doses, which we know is a convenience factor for patients. SANRECO is designed to start at pharmacologically active doses. So we hope then to easily, or quickly, I should say, identify an optimal dose to move rapidly into phase 2. PV patients may benefit through a reduction of phlebotomies and increase in iron stores, which could lead to a reduction in fatigue, which is an associated symptom. SANRECO is now open to recruitment in the US. And to find out more about that, you can look at clinicaltrials.gov, and I have the list here, or you can reach out to us at Silence Therapeutics. With that, I'll stop and thank you.
Thank you very much. It's just so great to see all of these companies doing such fine work. You know, in the 20 years I've been in this space, this is the most activity I've ever seen. So I know our patients in the MPN community are very, very excited. Thank you very much. And I'm sure we'll see each other again soon.
I hope so.
So we'll move on to protagonist, Dr. Afodi Ajari, who is the Executive Director of Clinical Development Are you with us yet? I know we are just connecting with him. Oh, okay. And so in the meantime, I do have a question for GSK. If you are still with us, Jessica?
Still here.
Okay, I will be 90 in a few months, and I have had PV since 2005. I was first on hydroxyurea, then Jakafi since 2000 and this looks like 18, and am doing well. Am I too old to join the clinical trial?
That's a great question. I can refer you to our medical team to answer that. They would be the ones that would have more information on our clinical trial inclusion and exclusion criteria. Let me get that and I'll maybe put it in the chat what that website might be for you to get further information on that.
Yeah, and that's a great question for
Yeah.
everybody, because even though many of you showed your inclusion criteria and exclusion criteria, I do think very often people that are older, I mean, she'll be 90 in a few weeks. Now, when my mom was 90, she was still so with it and active and, you know, doing very well. But I know there are age limitations on many, many trials. I'm not sure about 90, unfortunately, but anyway, thank you.
And
Jessica, for answering that.
and I'm sorry, just to add one more thing, the Momentum trial that we mentioned previously, that is looking at myelofibrosis patients. I believe PV patients, primary PV patients were actually excluded.
Right.
So that specific trial probably wouldn't be the right one for the patient that asked that question. But again, I'll get some information on where they could get further information on all of the clinical trials looking at momelotinib.
Right. And, you know, this is why we host these programs, because so many patients are listening and, you know, many want to participate in a trial, but there are some barriers to that. And sometimes it is their age, unfortunately. But anyway, we will see if Dr. Ajari, are you with us yet? No? Okay, is Virginia here yet? We have two more presenters that apparently are not quite with us yet. Now, we are running very quickly through these presentations. So Nora Boyer has raised her hand. Let's see what Nora has to ask. We did have some questions about location. I know many of you had your links to where the trials were taking place. If you could, we'll look back in the program as well, but I would like to form a comprehensive list of all of the trials. I think we have them on our website as well, but something very quickly that our patients that are on these programs, on these webinars, can access very quickly. And so we will go through and look at those, but if you have that slide and can just send us the slide with your information, that would be fantastic as well.
Hi, I'm on the call here. Can't you all hear me?
Is this Dr. Ajari?
Yes, I've actually been on the call, so
Okay.
I have the ability to share my slides.
Yes, if you want to share your slides, this is Dr. Ajari, who is the Executive Director of Clinical Development at Protagonist. And are you able to share your screen?
Right now, yes. So let me do that now. Thank you.
Okay. Because I don't see you anywhere yet. Oh, there you go. Okay, I'm going to
see my screen.
There it is. I'm going to leave so you can present, okay? Thank you for joining us.
Thank you. Thank you all very much. Thank you for the opportunity to have this discussion today with you. My name is Ifodi Ajari, and I'm the Executive Director of Clinical Development within Protagonist. Protagonist is a biopharmaceutical company located in the South San Francisco Bay Area. We're a strong biotech company with a bit over 120 employees. Our platform is based on a proprietary peptide technology with rusfertide, or what we call PTG-300. It's our lead investigational drug candidate. Rusfertide is a synthetic mimetic of the naturally occurring hormone hepcidin. And as you know, hepcidin regulates iron metabolism and also controls the absorption, the storage, and distribution of iron in the body. We are excited to announce that our phase 2 REVIVE study that a good number of us are aware of, which compared rusfertide to placebo in 70 PV patients, was recently unblinded and yielded positive results. And I'll be showing you the slides very shortly. As a result, we have moved forward with a phase 3 trial for patients with polycythemia vera. This we call the VERIFY study. It has already begun and we are currently enrolling. Our target is to walk through 100 sites globally and looking to enroll 250 PV patients. This VERIFY study allows both high-risk and low-risk PV patients to enroll, and it's considered an add-on trial. In other words, rusfertide is added on to the normal standard of care. It is a randomized placebo-controlled trial, and evaluating the efficacy and safety of a once weekly subcutaneously self-administered dose of rusfertide in PV patients. We have a website that, if someone has PV, you can answer a number of questions, up to 9 questions on this website. It will help determine if you qualify for our study. It will also reveal to you the different cancer centers that have the study open near you within up to 150 miles. The website is www.polycythemiavera-clinicaltrial.com. All right, based on the analysis of real-world data by Dr. Vestervek, we all learned that about 22% of PV patients have consistently controlled hematocrit. And for the rest, we conclude that they all have uncontrolled hematocrit levels. So data by Dr. Macioli shows that for these patients with uncontrolled hematocrit greater than 45, they're all predisposed to a greater risk of thrombotic events, cardiovascular events, and this actually may impact their quality of life. This slide is just to quickly have a recap of how hepcidin is central to the regulation of iron metabolism. Its effect on a cellular level involves binding ferroportin, which is the main iron export protein, resulting in its internalization, as you see, and degradation, and then leading to iron sequestration within these ferroportin expressing cells. So if there's any aberrantly increased hepcidin, it brings about systemic iron deficiency with or without restricted erythropoiesis. For our drug rusfertide, it potently functions as that hepcidin mimetic by targeting this imbalance of iron metabolism, targeting ferroportin, thereby working in that same manner. So just like hepcidin, our drug would induce endocytosis and proteolysis of ferroportin, thereby decreasing the delivery of iron to plasma. So through its effect of helping to restrict the availability of iron, for the red blood cell formation, rusfertide may really be an effective therapy for these conditions where there is excessive erythropoiesis, like polycythemia vera. We have about actually 3 studies, 2 phase 2 and 1 pivotal phase 3 study that I already just referenced. The REVIVE study, which is a phase 2 study that I'm going to talk about very briefly, had data that we've actually presented. We presented last year at EHA, then again at ASH later on in the year. There was a specific study for those that have really high hematocrit level, and we also have the VERIFY study that has actually started enrolling. For this phase 2 REVIVE study, It was conducted in 3 parts. There is the part 1A, as you see here. The part 1, which is a 16 plus 12, that is a 28-week open label dose finding phase, beginning from the first week and ends at the beginning of week 29. This period being made up of a dose finding as well as an efficacy evaluation part. It entails dosing patients with or without 4 weekly titrations to maintain their hematocrit to less than 45%. The second part is a 12-week blinded 1-to-1 fixed active versus placebo randomized withdrawal phase over a period of 12 weeks. Now that begins at the week 29 time point, as you see here, and ends at week 41. Part 3 is the 3-year open label extension period. Now, at the time of generating the data sets I'm about to show you, 10 subjects have been in this open label period for at least 2 years. Over 30 patients have been on it for over 18 months, and way over 50 have been for at least a year. The endpoints that we have been targeting are safety, maintaining hematocrit at less than 45, reducing phlebotomies, and assessing the effect of rusfertide on the modified NPN symptom assessment total symptom score form. So this is what we're showing you here, that our drug actually met the primary endpoint of efficacy with a p-value of 0.0003. 62% of the subjects that were on rusfertide, shown in orange here, versus 18.5 on placebo, which is great, are shown to be responders, highlighting very significant and very frankly remarkable efficacy in favor of rusfertide. Conversely, let's note that for the 8 non-responders in the rusfertide arm, 3 still fulfill the phlebotomy eligibility criteria and the endpoint we're targeting. The remaining 5 discontinued study intervention during this period, but all the 8 non-responders continued in the part 3 open label extension, and 7 out of 8 are currently still continuing rusfertide. So 92.3% of the subjects in the rusfertide arm did not get any phlebotomy during the 12-week randomization part of the study, which was that second part I had showed earlier. Now, using the same criteria of phlebotomy eligibility, 92.3% of the subjects, as I said earlier, in the rusfertide arm did not get phlebotomy during the 12-week double-blind efficacy period. Now, when you compare with the preceding dose titration and open label efficacy periods from the part 1, which were 76.9 and 87.3 respectively, we still show increasingly consistent effects on them being phlebotomy free, bearing in mind that even in this part 2 period, we allow them to dose adjust. So these results are really quite compelling, and we are really excited to present them. These curves were put here to really show those participants that have the highlighted outcomes of being called responding to treatment, or people that really experienced an absence of phlebotomies during the period they were studied, and those that consistently kept their hematocrit levels at less than 45% based on taking rusfertide. We show very clearly that over the period that we studied them, looking at the horizontal axis here representing time in weeks, and the vertical axis showing the percentage of those participants with the outcome of interest, either as tagged being responders or people that experience an absence of being phlebotomized or consistently keeping hematocrit at less than 45, we really saw very impressive results across the board. In addition to rusfertide shown here demonstrating strong efficacy, we also see that it works very fast. We see the onset between 4 to 8 weeks, and the effect is sustained, with very impressive p-values, 0.001, 0.001, 0.001 across the board. Then we looked at the part 1 for those that really got the open label drug as a way of looking at whether we are able to very objectively and clearly evaluate symptom improvement. The effect of rusfertide on the modified MPN symptom assessment form total captured very clearly the symptom improvement, very clearly, across different domains that we studied, from the point of looking at the problems with concentration, itching, fatigue, inactivity. The results stayed really consistent, and we're glad to present that. Okay, going to the phase 3 pivotal study, I would like to just give a summary of what this study is going to be about. I had called it the name Verify study earlier, and it's really on, one of the reasons why we're here today to really have this discussion. It's ongoing. It's a phase 3 pivotal study. We are looking at 250 subjects, capitalizing on the successful outcome of the data from the phase 2 Revive study. It's in 3 parts. Part 1A is a randomized double-blind placebo-controlled add-on parallel group period for 32 weeks. These subjects will be randomized in a 1-to-1 fashion, blinded, for 32 weeks of having rusfertide as add-on or giving them placebo. Now, these patients would be expected to have ongoing therapy for PV, because randomization is going to be stratified by their ongoing PV therapy. The second part, which is part 1B, is the open label treatment phase, where, for those that have completed part 1A, they will successfully receive rusfertide for 20 weeks. That is from week 32, as we see on this slide, through week 52. The third and final one is what we call the part 2, which is a long-term extension period for 104 weeks, during which all the subjects that complete part 1B will continue to receive rusfertide. Our primary endpoint really is to look at the proportion of the subjects that are achieving a response starting at week 20 through 32 inclusive, that are getting rusfertide compared to placebo, as shown here using the dotted lines. A response is defined as absence of phlebotomy eligibility. And if you think about phlebotomy eligibility, for anyone that, based on NCCN guidelines, that has a confirmed hematocrit of greater than or equal to 45%, or that we say has at least 3% higher than their baseline hematocrit, being that value immediately prior to randomization at week 0, would say, okay, those persons are phlebotomy eligible, or anyone that at any point has the hematocrit greater than or equal to 48% regardless of treatment. And some of our key secondary endpoints will be to look at the average number of phlebotomies, between weeks 0 and 32 inclusive, and also looking at different proportion of those subjects with all their different hematocrit values sustained below 45%. This is just a summary of some of the things that we have talked about, that for those patients that would always require frequent phlebotomy with or without their baseline cytoreductive treatment, we've been able to demonstrate, for at least 2 years in the Revive study, that they remained essentially phlebotomy free. And with the additional studies that we are conducting, we have very promising reason to believe that we are onto something with our study drug rusfertide. These are really the potential attributes based on our learnings. It is clearly an iron regulator, and we'll be able to address a broad spectrum of PV patients, both high risk and low risk. Then there's the option of being able to dose adjust. That flexibility affords us the ability to modulate how we gauge their hematocrit levels with the dose of rusfertide that they are giving. The onset of action, as we used one of the slides to demonstrate, is really very quick. In a matter of weeks, we start seeing hematocrit levels going down, and there's sustained hematocrit control, there's continuous control, and across the board for the different domains with respect to symptomatology, we see symptom improvement. On that note, I'll stop presenting. Happy to take questions, but would like to quickly just mention that for those willing to join, we have a portal where you can go in, www.polycythemia-vera-clinicaltrial.com as a place to indicate your interest, and you'll be able to get connections to any site that is close enough to you. Thank you.
Thank you very much, Dr. Ajari. That was excellent, and I appreciate you being here with us. We will move on to AbbVie and Dr. Virginia Tajero, who is the Associate Science Director. Are you with us, Virginia?
I am. Hello.
Thank you. And I appreciate you coming early. I know you had some other scheduling issues, and this program has just been flying by. So we appreciate you being with us.
Absolutely. And it's my pleasure. Can you see my screen?
I can.
Okay. So you just see the slides, right?
I'm sorry. If you put it in present mode, we will just see your slide.
Okay. Can you see it?
Yes, there you go.
Okay, absolutely good. Okay, so hi, Anne. Thank you, and thanks to MPN Advocacy and Education International for allowing me to discuss AbbVie's clinical myelofibrosis program today. My name is Virginia Tajero, and I'm an Associate Scientific Director in U.S. Medical Affairs at AbbVie. Okay, so we are currently evaluating navitoclax in myelofibrosis, but please note that navitoclax is an investigational agent and is not currently approved by the FDA or other global regulatory health authority, and safety and efficacy are still under evaluation. AbbVie is a global pharmaceutical company and a leader in the transformation of care for patients with blood cancers. We are striving to advance standards of care and bring transformative treatment options to patients, including those with myelofibrosis. So navitoclax is a first-in-class oral small molecule that binds to two proteins called BCL-XL and BCL2, which are pro-survival proteins that prevent cells from dying. Cancer cells can hijack the machinery that makes these survival proteins to keep themselves alive. So navitoclax binds to BCL-XL, which can cause myelofibrosis cells to die. JAK inhibitors shut off the pathway that increases production of BCL-XL, making myelofibrosis cells susceptible to BCL-XL inhibition. So given the results seen in the phase 2 REFINE study, AbbVie has proceeded with 2 phase 3 studies in the fall of 2020, both evaluating the combination of navitoclax and ruxolitinib in myelofibrosis in a larger patient set. The first study, TRANSFORM-2, that's the second bucket, includes patients with myelofibrosis who have not received prior treatment with a JAK inhibitor and is evaluating the safety and efficacy of navitoclax combined with ruxolitinib versus placebo and ruxolitinib. Now, the second phase 3 study, TRANSFORM-2, is currently seeking more patients to participate. This study includes patients with myelofibrosis who have worsening disease as evidenced by an enlarged spleen and symptoms despite previous or current treatment with a JAK2 inhibitor. TRANSFORM-2 is an open label study, meaning patients and investigators will know what agent they are receiving, and will evaluate combination of navitoclax and ruxolitinib versus the best available therapy, which is at the discretion of the physician and could include ruxolitinib, fedratinib, among other options. We currently have more than 30 sites across the US for each study, and TRANSFORM-2 is still seeking patients to participate. Here you can take a look at eligibility criteria and study endpoints. And for more information regarding the ongoing studies, you can take a look at www.myelofibrosisresearch.com. The site includes further information about each study, a questionnaire to determine if you might be eligible to participate, and a map of current site locations. I thank you very much for your time.
Okay, well, thank you for that. We really appreciate it. And as mentioned, this has just flown by. We end up getting more questions after the fact. And when we send this out tomorrow, probably the recording will be ready to be sent. But I so appreciate all of you. We could not do our work without your support. And I'm just absolutely flabbergasted at how much is happening in the MPN space. And I appreciate everything you're all doing, every single company. And we look forward to seeing you soon. We'll be in San Diego next week. I think some of you are joining us. And then perhaps some of you at ASCO will see. Thanks again and have a great day.